So in the previous class, we had talked about the Human Genome Project, in which we saw how the DNA is taken out, then the DNA is cloned, and after that, what happens to the DNA? It is sent to the DNA sequencer, so as to process and sequence the DNA molecule. After the whole entire thing was done, the salient features of the human genome were noted. And right here on your screen are the salient features. So first of all, the human genome consists of 3,164.7 million nucleotide bases, meaning those adenine, guanine, thymine, cytosine, these are that many in our, in the human genome. The average gene consists of 3,000 bases, okay, but the size vary greatly. With the largest gene is dystrophin, which has 2.4 million bases, and TDF gene, it is another gene, the name of the gene, that is the smallest gene that only has 14 bases, and that's it. This was the information that was, found after the Human Genome Project was done. Then a total number of genes is estimated at 30,000, much lower than the previous estimate. So the total gene in the human itself is 30,000, which is less than the previous estimate of this 80,000 to 1,40,000. And this signifies, or this tells us, or this has cleared the fact that 99.9% of the nucleotide bases are exactly the same in every individual, whether it is you all and me. We all have 99.9% bases very similar to each other. We just differ by that 0.1%. That you are like you and I am like me. It's because of that tiny, tiny difference. And that difference is actually really huge. But if you think about us, like all the proteins and all the enzymes that are made in my body, they are also made in your body. All the, you can say, translation that happens in my body, it happens in your body also. So it's like basically everything is same. Just the fact that that 0.1% or that is the how we are different from each other. And the functions are unknown for 50% of the discovered gene. We still don't know their functions. Less than 2% of the genome codes for protein. Imagine out of that 99, or you can say the whole entire genome, that is 30,000. Out of that, only 2% will be coding for the proteins in our body. And then our whole chunk of our genome is made up of the repeated sequence. It is repeated n number of times, like repeated throughout our body. Now, repetitive sequences, these are the stretches of DNA that are repeated many times, sometimes hundreds to thousands of times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. That's what they do. Now, chromosome number 1, it has most number of genes, that is 2968. Imagine 2968 genes are present in our first chromosome number. And the Y chromosome, which is the sex chromosome, that just has 231 genes. Now, the scientists, they have identified about 1.4 million locations where single base differences occur in humans. Just the difference of 1 or 2 bases. This is known as SNPs, that is single nucleotide polymorphism, which is pronounced as SNPs. So this information tells us that in majority of our, you can say, genome, every single thing is the same. It's like the point differences that makes us who we are. And our genetic sequence or our genome has been repeated several thousands and hundreds of times. Clear, students? So the features of the Human Genome Project, like what is the conclusion that they came to? Is that clear to everyone? Any doubts? Oh. Now, the completion of the applications and future challenges. This is something that they'll not ask you, but you should know the things about it. So the completion of first phase of Human Genome Project has been compared to the discovery of antibiotics, because it has opened a vast database of knowledge about various aspects of human genome. Soon, we shall be mapping all the human genes and all the sequences, the transposons, and even the junk DNA. This is the aim. Then there are more than 1200 genes that cause common cardiovascular ailments, meaning those diseases that are related with the heart. Then endocrine diseases like the diabetes, Alzheimer's, which is the disease, like it's a neurological disease, cancer, and all of that, right? So if we know the genome that undergoes replication and causes that disease, then we will be able to treat it also, isn't it? We will be able to not let that genome get mutated and cause any kind of ailment. Single gene defect produces a number of hereditary diseases. Hereditary diseases meaning those diseases that are passed on from one generation to another. So that also happens. And if we know the genome and the sequence and everything, then that can also be correct. It will be possible to study interaction between genes, proteins, the mechanism of tissue forming, organ forming, all of these things we can actually find out, or we can actually know about with the help of the Human Genome Project. If it is done furthermore, also carried on. And then it holds the promise of healthier and longer living, designer drugs, genetically modified diets according to the need of individual human beings. So, because of all of these things that were, initially done, today we know the sequences, we know the base pairs and the whole entire, you can say, coding mechanism that happens in our body. And because of that, there can be like a lot of research that can go on and everything related. So, is the Human Genome Project clear to you, students? Any doubts anywhere? Okay. Okay,
No, ma'am.
everyone please switch on your videos and keep it on. I'll let you switch it off while you write. Alright, so now coming on to the last topic of this particular chapter, that is DNA fingerprinting. Now DNA fingerprinting, it is a technique like how you people might have you seen CID in TV shows like that? Where the crime investigation kind of TV shows. So you might have seen that whenever there's a crime scene, people start to collect the specimens from that area. If there's a blood stain, people collect that. If there's a hair lying around, they take it and they collect that. Right? Why do they collect? Do you people have any idea as to why it happens? Like, have you people seen those kind of TV shows? The crime related one, the people solve, like they investigate who is the murderer or who is the criminal over there, right? So why do they collect those things? Does anyone have any idea about that?
They'll be able to recognize the DNA and who is
Mm.
a criminal.
Yeah. See, normally, whenever there is any kind of criminal case or any kind of investigation that has to go on, there are suspects, isn't it? People suspect that, okay, this might be the person who does it. This might be the person who has done this. So, what is that one thing? Like, normally, if you see, normal fingerprints are also taken, right? And with the matching of fingerprints, you can find out that who is the person that opened the door and stuff, right? Furthermore, if you think about the stuff like this thing, blood, think about the stuff like saliva or the hair strand or anything of that sort, just remember that all of these things, they have our DNA present. They have the DNA. So if you do DNA fingerprinting, you can match the DNA and that will be the most accurate result. If the DNA matches, like some people can fabricate fingerprints, isn't it? You might have seen that if you put, I don't know if that's true or not, but if you put some sort of latex or something there and you put your fingerprint and then another person can use that latex to put your fingerprint elsewhere because these are the lines that can be marked. But you cannot manipulate DNA. It's not possible to manipulate DNA. So it's like the most accurate test there is. So that is what DNA fingerprinting is about. So you find out which individual does the DNA belong. So the DNA fingerprinting, it is a technique used to determine nucleotide sequence of certain areas of DNA which are unique to every individual. Now, obviously, if you were to study my DNA and you were to study your own DNA, it would be 99.9% same in both of our cases, right? It's that 0.1% that will make me the criminal or make you the criminal, isn't it? Huh. So how should I make you the criminal? So I will go for the gene that differs. I will go for the gene that makes you different from it, right? So DNA fingerprinting can distinguish one human being from another with the exception of monozygotic twins. What is monozygotic twins, students? Monozygotic twins. Anyone?
Like twins from the same zygote.
So, in what do you call them in the layman language?
Mm.
Identical twins. Have you heard about them?
Yeah.
Those two people that look exactly the copies of each other, right? So why do they look like exactly like each other? It's because when the zygote was formed as a result of fusion of the male and female gamete, this zygote split into two parts and then both the parts began to develop into embryos. So that is what monozygotic means. Very good, Jesslyn. Mono means one, zygotic refers to the zygote. So those people that develop from the splitting of one zygote. Okay. So in their case, because they are formed from all of their DNA sequences are same. It's like if one of the twin commits a crime, the other twin can also get caught because their DNA sequences are exactly like each other. So for them, this thing is not useful. But 99.9% base sequence among humans is the same. We all know that. That 0.1% of the genome or 3 into 10 to the power 6 differences in the base sequences that makes us who we are. And the differences occur not only in genes but also in repetitive DNA. So it's not just found in the genes but those DNA sequences that are repeated multiple times. So DNA fingerprinting, it involves the identifying differences in some specific regions in the DNA. And which region? So repetitive DNA. So these repetitive DNA, they are separated from the main or the bulk genomic DNA as different peaks during density gradient centrifugation. So when the centrifugation of those DNA strands are done, so the one that is the bulk genomic DNA, meaning the core DNA, that is separated different. Okay, and the repetitive DNA, the 0.1% one, they are separated different. So the bulk DNA forms a major peak in the centrifugation, whereas others, they small peaks are formed, and we call them as the satellite DNA. So this satellite DNA is what we study when we want to study the DNA fingerprint. Is it clear? So satellite DNA, it is classified into following categories on the basis of the base composition. Base means the nitrogenous base, adenine, guanine, cytosine, thymine. Then the second thing is the length of the segment and the number of repetitive units. So according to these three criteria, the satellite DNA are of two types. VNTRs and then SSR. Now, VNTR means variable number of tandem repeats, or you can call them as mini satellites. So they are surrounded by conserved restriction sites. A small DNA sequence is arranged tandemly in many copy numbers. So it is like repeated several times. The copy number, it varies from chromosome to chromosome. So that like the number of repeats show very high degree of polymorphism. So it varies from 0.1 to 20 kb. So that much. Okay, so the VNTR is the first thing that comes under the satellite DNA. And then there is single sequence repeats or short tandem repeats. They are called as microsatellites. They just have 1 to 6 base pairs. These sequences, they are not coding for any protein. They are not the coding sequences. It's just that they make a huge bulk of the human genome and they show polymorphism. Now, polymorphism means that they will be existing at various places you can say at various genetic level. So since the DNA from every tissue, such as the blood, the hair follicle, skin, bone, saliva, sperm, all of these, they show the same degree of polymorphism, meaning they exist in different forms. Okay, and these are inheritable, meaning they can be passed on from the parents to the generation. So this DNA fingerprinting, have you people heard about this thing, paternity testing? To find out who the kid belongs to. So that is also done with the help of the DNA fingerprinting. The DNA is matched in the case of disputes. So as polymorphism in DNA sequence is the basis of genetic mapping of genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means. Polymorphism means the variation at genetic level, meaning that same DNA existing in different forms at different levels, and that is because of mutation. This is what, in general, polymorphism means. And this polymorphism is the basis of the DNA fingerprinting. So if more than one variant, that is allele, at a locus occurs in human, allelic sequence variation has traditionally been described as DNA polymorphism. If more than one variant, meaning more than one allele, it occurs in a human with frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, then we call it as DNA polymorphism, meaning it is occurring at every level and in the whole entire population. So we say that this DNA polymorphism is higher as compared to the rest. Clear?
Yes, ma'am.
Hmm. So the probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation in these sequence may not have any immediate effect in the individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of the polymorphism. Because of mutation, polymorphism arises. There is a variety of different types of polymorphism that starts from single nucleotide chain to large scale change. Okay, and even for evolution, polymorphism plays a very important role. So how do we do the process of the DNA fingerprinting? So the basis is VNTR, which is variable number of tandem repeat, that shows a very high degree of polymorphism. And this, remember the person's name, like who developed the DNA fingerprinting. So it was Alec Jeffreys. He was the one who developed this whole entire technique. Now see students, like for identifying or for separation, like for example, what are all the biomolecules you can think of? You can think of DNA, you can think of RNA, and you can think of protein, right? Now for the separation or for the identification of different types of these things, uh, molecules or biomolecules in general, what do we do? Basically, when you want to transfer them, so you need different blotting techniques. Okay, blotting techniques, just like you might have heard about oil blotting sheets that people use on their face that picks up the oil, right? So same way, when you are cutting DNA, when you're cutting protein, there is a membrane that comes which you can put and take the DNA on it, like that, if you imagine. So you have to remember which one. So DNA, RNA, each of them have different this thing, blotting techniques. So for a trick I'll give you, you'll write snow and you'll write brown. Okay, then you'll cross the O's. So southern blotting is for DNA, northern blotting is for RNA, and western blotting is for protein. So these are the different blotting techniques used for the different biomolecules. So you always use this snowdrop technique, you'll always remember. Okay, so snowdrop, S stands for southern blotting, that is for DNA. N stands for northern blotting, that is for RNA. And W stands for western blotting, that is for protein. So now let us see the steps. Firstly, whatever DNA you want to study or whatever DNA you want to do the fingerprinting of, you take that DNA, you isolate the DNA. So DNA isolation is the first step. When you take the DNA, now you people also know that DNA is a really, really long molecule. So we can't study that long of a molecule all at once. So what do? You basically cut the DNA into several tiny, tiny fragments. Okay, so that it is easier for you to study. So that is called as digestion of DNA, and it is done with the help of restriction endonuclease. So these are the enzymes that helps to cut the DNA into pieces. Then Once you're done, you've made pieces. Now you want to separate those pieces because you want to take one to study, isn't it? You take minute things to study. So you want to separate those DNA pieces or fragments. So that is done with electrophoresis technique. All of these techniques you will be studying in biotechnology chapter in depth. For now, you just understand that electrophoresis is done. Then you transfer them to synthetic membranes, the blotting techniques. So if you are doing it for DNA, you do southern blotting, isn't it? So you do the blotting of the separated DNA fragments to the synthetic membranes such as nitrocellulose or nylon membrane. Then you do hybridization using labeled VNTR probes. So whatever DNA you have selected, you sort of label it, okay, with a probe. And then you detect the hybridized DNA fragment by the method of autoradiography. And it will give you different bands. Like once you are done doing it, different bands will be there. So you can make this whole entire thing for two individuals and then you can match the bands. Like meaning the blood you have discovered from the crime scene and the person you are suspecting to be the criminal. You take that person's blood and you do the same procedure for both of them. You'll receive bands and you can analyze. Understood? Also, remember this thing that after hybridization, the autoradiograph, which is the machine, it will give you different bands of different sizes. And this will give you the pattern for individual DNA. The sensitivity of the technique has been increased by the use of PCR. PCR is polymerase chain reaction. Meaning, if you take one single DNA and you do PCR of that, you can make 1 billion copies. PCR is this tiny machine. It's like a box. But with the help of that machine, you can make thousands, billions and billions copies of DNA. So DNA, even if you get a tiny bit of DNA, like half a hair strand, little bit of the saliva, that is more than enough because you can increase that. For example, you went to a crime scene. Why am I saying crime? But see, let's just imagine it like that. You went somewhere. The crime that happened in that area has become very old. So now there will be no fresh DNA thing for you to get, isn't it? So even if you get a tiny bit, let's just say you got a tiny speck of blood, let's just say tiny speck. So if you use that, if you isolate DNA from that, and then you do PCR of it. So now instead of having that one speck, you have 1 billion copies of it, billion if you need it. So because of that, efficiency has been increased. That even with a tiny amount, you can do a lot of things. So that is what PCR has made easy for. And like this whole entire thing, it happens in a tiny box. So this is the pictorial representation. If you people get asked about DNA fingerprinting and if it is a 5 marks question, you should try to draw this. Okay, it's like all boxes only. So the first thing or the first step is taking the blood sample, then extracting the DNA from the blood cell. Then you cut the DNA by using restriction enzymes, and then you separate them using the gel electrophoresis. You blot them using the southern blotting because you're doing it for DNA. And then after that, the radioactive probing you do, make it radioactive in nature. And then what do? You put it for the radiography, autoradiography. And then when you like visualize the autoradiograph result under the X-ray, these band-like structures are what you're going to observe. And that is how it's going to tell you which one is like the thing, like how see here. Like the paternal chromosome, the maternal chromosome. Okay, these are matched like that. So what are the applications of the DNA fingerprinting? First of all, paternity-maternity disputes. In any dispute cases that the child belongs to whom, this is the perfect technique to find out. It's like a perfect result. Second, criminal identification and in forensic. For that, this is clear, students? Then it is also used for personal identification and close relations of intending immigrant. So for immigrants also, this can be used. So in general, to find out the connectivity, the DNA fingerprinting is almost the best technique there is. So with this, we have actually come to the end of the chapter. How are you people feeling it? Your very boring chapter is now over. Are you people happy? Happy. So it was boring. Eh? No, ma'am, you taught it so nice. It was not boring. No. Yes, it was boring. Very boring. Okay, now see. There are few things that you don't have to note down because it's all what's there in your NCERT. So you don't need to note down. Some things are important. I'll tell you what all things you have to note down. And today I'll be starting a new chapter. Okay. So first, you people write down the features of, I'm putting tick marks over there. Okay, so features of Human Genome Project, that salient features you have to write. This is important. These all things you don't have to write, none of the DNA fingerprinting, and then the steps and the diagram, this all. Okay, so this and the salient features, this whole entire big thing you don't have. Okay, so now you people can note it down, then we'll move forward. Done, students? Students, are you done till here?
Yes. Yes, ma'am.
All right. Done, students? Shall I scroll down? Well, students, then till here?
Yes, ma'am.
Okay, does anyone have any doubts up until now? All right. So with this, we are done with the molecular basis chapter. Now we're going to start with a new chapter today. And I was thinking of doing human health and diseases with you. Okay. So we will be beginning with human health and disease chapter. Is it okay, students?
Yes, ma'am.
All right. So human health and disease, this chapter is about all the different kinds of diseases and all the different kinds of pathogens that cause the diseases in us humans. Other than that, it also talks about the cycle that happens in the disease, and then what are all the things because of which a person can, protect themselves from the disease. What is immunity? What are the types of immunity? And all of these things we're going to see in this chapter. So today we're going to discuss the very basics of the very beginning. So the father of medicine, Hippocrates is known as the father of medicine. William Harvey, he was the person who discovered the presence of blood circulation in the body. Now, if we talk about being healthy, so being healthy is being, like being fit in terms of physical health, in terms of mental health, and in terms of social health. So when a person is fit in all these three aspects, along with the biological health, right? Then a person is said to be healthy. Then you can say that the person is healthy. Now, our health can be affected by multiple of, you can say, factors, right? Genetic disorders. Now, if a person is born with inborn error, inborn mutation, or if a person is born with the wrong kind of chromosome number, then we constitute that as the genetic disorder. No one knows how, whether or not it will be happening in the offspring or not, all of these things. So the first way the health could be affected would be because of the genetic disorders. Second, infections. Now, when a person is born and that person lives on the land for a period of time, right? Slowly, like every day we get exposed to the pathogen, we get exposed to the, you can say, all the infectious stuff that are there in our surroundings, right? So as a result of which, what happens? We get the infection, we get disease, and our health is compromised. And the third thing is the lifestyle. The lifestyle that we lead or the lifestyle that we have, that also has a great big impact on our health. For example, if you do exercise, if you eat healthy, don't eat enough junk food and stuff, then you will stay fit for a long period of time. But if a person is there who doesn't have a very active lifestyle, that person is very sedentary in the life, and the person eats a lot of junk and a lot of, a lot of highly oily food and processed foods and a lot of meat, like excessive amount of it, then also the person's health could be affected. The person's body physiology could be affected. So these three things, they affect the health in general. Genetic disorders, infections, lifestyle. Is it clear, students? Hmm? Yes or no?
Yes, ma'am. Yes.
Hmm. Moving forward, talking about what are the various things that a person can do to maintain the good health. The first thing is balanced diet. Now, you people know that in a balanced diet, all the components that are necessary for a human being, that's there. Whether it is proteins, whether it is carbohydrates, vitamins, minerals, necessary fat molecules, roughage, all of that is there, along with the water requirement that our body requires. So, balanced diet, personal hygiene, and regular exercise. These are the three things that are extremely important to maintain good health. Despite of having no pathogens, despite of having no infectious agents around you, if a person is not clean and doesn't practice cleanliness, then that person is bound to get ill one day or the other. So personal hygiene is that important, as well as the regular exercise. Is this much clear, students?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
Okay. All right. Now, you people, So this is like the basic introduction of this chapter. Now let's see what all things you people have to see further.